Good day, everyone, and welcome. This is Ray Ford with FDA Center for Drug Evaluation and Research, Small Business and Industry Assistance Program, also known as CEDAR SBIA. Thank you for joining us today for our presentation on the immunogenicity information and labeling. During this webinar, FDA will highlight why immunogenicity is important to consider for drug development, discuss the draft guidance for immunogenicity information in human prescription therapeutic protein and select drug product labeling content format, and also discuss creation of the new dedicated subsection 12.6 for immunogenicity information, immunogenicity subsection, in the clinical pharmacology section of labeling. The webinar will be posted in its entirety on our website within approximately 5 business days, and that website is fda.gov/CEDAR-SBIA Learn. Before we begin, I'd like to cover some details. As mentioned earlier, a copy for today's presentation is available on the left-hand corner side of your screen below the Q&A chat pod, where the beginning title states Immunogenicity Information. This activity is eligible for a by request only attendance certificate which supports continuing education by SOCR, RAPS, SQA, and ACRP. Please refer to our website at fda.gov/CEDAR-SBIA for more details. If you would like a certificate of attendance, please email your request to cdersbia@fda.hhs.gov. That's cdersbia@fda.hhs.gov. You'll be able to request the certificate of attendance only for 2 weeks, and that's until April the 19th. Additional tutorials are available through fda.gov/CEDAR-Learn. I'm also very pleased to announce that in addition to the certificate of attendance, which supports Cs for RAPS, SOCR, SQA, and ACRP, this webinar is offering a total of 1.5 credit hours to physicians, pharmacists, and nurses. Note that this is an entirely separate process from that of the requested certificate of attendance. The Cs are available to all real-time attendees who self-identified as healthcare professionals during the Adobe Connect registration process. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than that of a single supporting company. C faculty and speakers are required to disclose to the attendees when products or procedures being discussed are off-labeled or unlabeled use, and meaning that's not FDA approved, and any limitations on the information that is presented. We have a spectacular lineup of speakers. The planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to their attendance and complete the final activity via the CE portal, and that CE portal is ceportal.fda.gov. Final activity evaluations must be completed within 2 weeks after the activity, no exceptions. So in this case, the deadline for this webinar is April the 19th. I apologize in advance, but we're unable to make exceptions to that deadline. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in the loss of credit for this activity. NABP profile number should be the 6 to 7-digit profile number assigned by the CPE monitor, and your date of birth should be in the month, day format, such as 0405. Please do not provide your license number. When you're in the CE portal, please click the My Account tab and then navigate to the Edit Contact Information to verify that your information is correct. Physicians and nurses may then view and print a statement of credit. Pharmacists should log in the CPE monitor 8 weeks after the last session of the activity to obtain their CE credit. To summarize, attendees do have 14 days from the day of the activity to log in, complete the required CE evaluations, and attest to their attendance to claim credit. SBIA will issue the CE claim code to all attendees via email later today. Be sure to complete that CE session evaluation on or before April 19th. In addition to the above process, if an attendee would also like a certificate of attendance that may be used in support of RAP, SOCRA, ACRP, and SQA credits, please email your request to cedarsbia@fda.hhs.gov. Certificates of attendance will be mailed until the April 19th deadline. Again, we apologize in advance, but we're unable to make exceptions to the April 19th deadline. For your convenience, we've included QR codes to access SBIA on LinkedIn, Twitter, and YouTube. Scan the QR code with the camera on your cell phone. This will provide you with a link to SBIA on the platform for the QR code that you scanned. To outline today's event, the webinar in its entirety will run approximately 1.5 hours, which includes a Q&A session. And if you have questions now or during the webinar, please enter those questions into the Q&A chat pod. We'll answer as many questions as time allows. Our speakers for today's webinar include Eric Brodsky, MD, Associate Director, Labeling Policy Team, Office of New Drug Policy, Office of New Drugs, CDER, FDA, and Daphne Gwynn, PhD, Regulatory Health Project Manager, Office of Clinical Pharmacology, Office of Translational Science, CDER, FDA. Please join me in welcoming our first presenter for today's webinar, Dr. Gwynn.
Thank you so much, Ray. So today we're going to discuss immunogenicity information and labeling. I'll first begin by highlighting our disclaimers. The views and opinions expressed in this presentation represent those of the presenters and do not necessarily represent the official position of FDA. The labeling examples in this presentation are provided only to demonstrate current labeling development challenges and should not be considered as FDA recommended templates. So here's an overview of what we plan to discuss today. First, we'll answer these questions. What is immunogenicity and why should we care about it? Followed by how does FDA evaluate immunogenicity? In the second part of the presentation, we will discuss immunogenicity information and labeling. So the learning objectives for this first part of the webinar are first to define immunogenicity, followed by reviewing the role of immunogenicity in FDA regulation. I'll then highlight why immunogenicity is important to consider in drug development. And finally, I'll provide the resources about FDA's approach to immunogenicity, specifically discussing current FDA guidance with immunogenicity relevant information. So we'll begin by addressing what is immunogenicity and why is it important to consider in drug development. Immunogenicity is defined as the ability of a substance to trigger an immune response in the body. We can classify therapeutic immunogenicity into two broad categories. Desired immunogenicity, which we would think of in the case of wanting to elicit an immune response to a vaccine or an allergenic product, or undesired immunogenicity, which would be assessed for some biotherapeutic products such as protein products like monoclonal antibodies, and some small molecules such as peptide products, which are less than 40 amino acids. For most CDER products, the development of immunogenicity would be considered undesirable. Within the context of this presentation, the focus is undesired immunogenicity and will be defined throughout as the propensity of a therapeutic protein product or applicable drug product to generate an immune response to itself, a related structure, or product complex, and/or induce an immunologically related adverse clinical event. So in considering the range of these potential immunological related adverse clinical events, we first consider immunogenicity that may impact pharmacokinetics, pharmacodynamics, and/or efficacy. Within that, there may be no clinical significant effect. For example, after receiving treatment, a patient may develop an anti-drug antibody. Those antibodies may not be binding to a critical region of the product and have no impact on the drug's activity. But that having no effect may not always be the case. Undesired immunogenicity may result in altered pharmacokinetics. For example, the formation of anti-drug antibodies may alter a drug's bioavailability. The anti-drug antibody formation may lead to a decrease in the drug's exposure because the antibody is binding has resulted in a rapid clearance of the drug. Binding of an anti-drug antibody could also increase a drug's exposure by decreasing elimination. Pharmacodynamics and/or efficacy may be impacted. Decreased exposure due to the formation of anti-drug antibodies could also lead to a decrease in efficacy. There are also cases when the impact on pharmacodynamics or efficacy are independent of the impact on pharmacokinetics. Next, we also consider the potential immunogenicity impact on safety. The immunogenicity impact on safety can span a variety of potential immune responses. Formation of anti-drug antibodies can cross-react with a native protein or receptor. I'll provide an example of this in an upcoming slide. The product could also be neutralized by the formation of an antibody or immune complex. The presence or activity of a drug could be potentiated, and immune complexes could be formed. Patients may experience hypersensitivity reactions or cytokine release syndrome in response to therapy. An example of how immunogenicity may have an impact on pharmacokinetics and efficacy of a product, adalimumab, is shown here. The graph on the left shows weeks on the x-axis and median adalimumab concentrations on the y-axis. The top line represents patients that have tested negative for the presence of anti-adalimumab antibodies. The middle and bottom lines represent the patients that formed anti-adalimumab antibodies. The middle line represents patients that have a lower antibody titer, and the bottom line a higher antibody titer. As shown, these patients with anti-adalimumab antibodies had reduced median adalimumab concentrations. In those same patients, the impact on dropout due to treatment failure is shown in the graph on the right. These patients with anti-adalimumab antibodies showed a loss of efficacy and were more likely to drop out because of treatment failure. When considering the impact of immunogenicity on safety, a classic case is pure red cell aplasia, or PRCA, due to immunogenicity in patients treated with epoetin alpha. PRCA is a rare disorder that impacts the production of red blood cells, or erythropoiesis, that leads to a progressive severe anemia. This is due to almost complete cessation of erythropoiesis, and the blood reticulocyte count becomes very low. In patients, the hemoglobin concentration declines to a rate that corresponds with the normal decay of red blood cells. To avoid a very severe anemia and maintain an acceptable hemoglobin level, patients require a red blood cell transfusion. PRCA has been described in patients treated with epoetin alpha, with most cases reported in patients being treated for chronic kidney disease-related anemia. Treatment with epoetin alpha can result in the formation of a neutralizing anti-epoetin alpha antibody, which can also neutralize the endogenous protein erythropoietin, or EPO. Patients developing PRCA due to anti-EPO antibodies are typically been on epoetin treatment for 6 to 18 months. The schematic on the left shows the representation of a typical case of epoetin-induced PRCA. The red line shows the hemoglobin levels, and the blue line shows ferritin levels. As shown here, the hemoglobin levels rapidly decline with cessation of erythropoiesis by the anti-EPO antibodies, despite continued therapy or even increased doses of epoetin alpha. This would be diagnosed by taking a bone marrow aspirate, which would show the presence of anti-EPO antibodies, and the patient would require a red blood cell transfusion. Now that we've defined immunogenicity, it's important we'll talk about and the impact of pharmacokinetics, efficacy, and safety. We'll now discuss how FDA evaluates immunogenicity. Each of these factors can play a role in assessing immunogenicity risk. Here, they're broken into 3 categories: treatment, patient, and product factors. Treatment factors, these can include dose, route of administration, such as intravenous, subcutaneous, or oral, frequency of the dosing, is it given daily, is it given monthly, the duration of the treatment, how long the treatment will be received, specifically, is it a long-term treatment, a short-term treatment, will it be given to the patient indefinitely? The next category is patient factors. A patient's genetic background may have an impact. Patients that have certain HLA subtypes may be more likely to develop certain autoimmune diseases, therefore more likely to have an autoimmune response. The patient's disease state being treated, Any concurrent illnesses or medications can also play a role. As an example, patients with cancer may have immune system dysfunction due to their tumor downregulating their immune system or them taking immunosuppressive therapies. This is a state when the immune response is less likely compared to patients with autoimmune disease such as rheumatoid arthritis, because these patients are more primed for an immune response. It is important to consider the different indicated conditions in which a therapy will be given for, and whether immunogenicity can be extrapolated from one condition to another. Finally, we have the product factors. These may include the size of the product, the protein structure or sequence, protein modifications, the target or biological activity of that target, the formulation of the product, contaminants or impurities that are found in the formulation, or any presence of aggregation. Assessment of all of these factors can help to determine the overall potential immunogenicity risk. So the next question is how FDA evaluates immunogenicity. Typically, immunogenicity is most commonly measured in clinical trials by assessing the formation of anti-drug antibodies, which are measured as a percentage of patients that develop anti-drug antibodies. This is done using a multi-tiered approach that is discussed in detail in the referenced guidance. A sensitive screening ADA assay is initially used to assess clinical samples. The screening assay should be validated, sensitive, and designed to detect low levels of low and high affinity anti-ADAs. Patients that are found to be ADA positive go to the confirmatory stage. For this stage, the sponsor confirms that binding of the antibodies is specific to the therapeutic protein product. Patients that are then confirmed to be either ADA positive or negative. This provides the ADA incidence, which informs the immunogenicity risk assessment and is included in the product labeling. Once the formation of antidrug antibodies is measured in patients, next, the impact of those antidrug antibodies should be assessed. And a question that comes up is, does the presence or amount of that antidrug antibody have a clinically meaningful impact on the drug's pharmacokinetics or patient response, including pharmacodynamic safety and efficacy? This would be assessed by evaluating if there are differences in drug concentration, treatment response, or safety-related events in patients with antidrug antibodies compared to those without. This would also be done over time. The treatment, patient, and product factors, along with antidrug antibody incidence and assessment of immunogenicity impact on PK, efficacy, and safety, are all considered when defining the overall immunogenicity risk for a product. So FDA has published several guidances to assist sponsors in assessing immunogenicity. Listed on this slide are two foundational guidances highlighting the agency's current thinking on immunogenicity assessment and assay development and validation. The first guidance discusses product and patient risk factors that may contribute to immune response rates, as well as risk mitigation strategies. The second provides considerations for development of and validation of immunogenicity assays for the detection of antidrug antibodies. Immunogenicity is also highlighted in the biosimilar and interchangeable biosimilar product guidances. The determination of biosimilarity includes a comparison of clinical immunogenicity, and in demonstrating interchangeability, a switching study may be performed to evaluate if switching between the reference product and the proposed interchangeable results in a difference in immunogenicity. FDA has also published guidance pertaining to immunogenicity potential for certain generic drug products. The FDA guidance on immunogenicity-related considerations for low molecule weight heparin discusses an alternative approach to assess the effect of certain changes on a product's immunogenicity risk. And the 2021 guidance on ANDAs for certain highly purified synthetic peptide drug products of rDNA origin discusses identification, reduction, and mitigation of risks related to peptide-related impurities, which can include the immunogenicity risk. With that, I will conclude this part of the presentation and turn it over to Dr. Eric Brodsky to discuss immunogenicity information and labeling. Thank you all for your attention, and I look forward to your questions. Eric?
Great. I am going to do one more sound check to make sure that folks can hear me. Great. Thank you, Daphne. So this part of the presentation, I'll be talking about when to and how to include immunogenicity information in labeling. So we'll discuss creation of a specific dedicated labeling subsection, subsection 12.6, called the immunogenicity subsection. This subsection is in the clinical pharmacology section of labeling, in section 12. The bulk of the presentation is describing how to develop the immunogenicity subsection, subsection 12.6, and then I'll go on to when to and how to describe and incorporate immunogenicity information in other sections of labeling, particularly clinically oriented sections of labeling, such as the adverse reaction section of labeling, the warnings and precautions section of labeling, and the clinical studies section of labeling. And finally, I'll provide some recommendations about when to update immunogenicity information in labeling. So the immunogenicity labeling guidance was published in draft about 60 days ago. Below, I have included the docket, a link to the docket. So far, as of last night, we received 2 comments from 2 different organizations. We really appreciate your comments. They were very thoughtful comments. You're welcome to include, folks are welcome to comment on the docket. We do look at your comments very thoroughly prior to finalization of the guidance. So the main purpose of the guidance is to assist FDA staff and applicants to incorporate immunogenicity information in labeling in a consistent manner. And this is particularly the purview of the guidance are therapeutic proteins regulated by CDER and select drug products that have immunogenicity assessments, such as certain heparins, peptides, and oligonucleotides. So one of the main purposes of the guidance is to differentiate products that have clinically significant immunogenicity, meaning that anti-drug antibodies have a clinically significant impact on safety and or effectiveness, and those that do not, where the anti-drug antibodies are not associated with any clinically significant effects on pharmacokinetics, pharmacodynamics, safety, and or effectiveness. And we'll talk about in later slides how potentially the guidance does this. So first, FDA did an examination of how we've incorporated immunogenicity information labeling. This was led by my colleague Daphne and other folks at the FDA. So we looked at, over a 5-year period, we looked at novel products approved by CDER. These are new molecular entities that had immunogenicity assessments, as well as therapeutic proteins approved in CDER. So there were 71 products, a total of 11 small molecules regulated under NDAs, and 60 therapeutic proteins during this 5-year period. And we found that almost all of the labeling included immunogenicity information in Section 6, the adverse reaction section. Now that is potentially problematic in the sense that the way we've defined adverse reactions according to our regulations are untoward events that have a possible causal relationship to the drug. And in many situations, immunogenicity anti-drug antibodies are not adverse reactions. They're not associated with safety concerns. So that was one of the things we wanted to potentially address in the guidance going forward. Secondly, one of the other findings of the guidance is that although 70% of labeling included an impact statement on safety and or effectiveness, 30% did not. So here I'm defining impact on safety or effectiveness if the labeling had a clear statement that there was an impact or a potential impact, or we didn't know there was an impact, or after a determination there was no clinically significant impact. So the challenge here is that sometimes we have labeling that report immunogenicity incidents, or maybe NAB neutralizing antibody data, but there's no information about the clinical meaningfulness of this. So one of the things that the guidance tries to do is encourage statements about the clinical meaningfulness of immunogenicity. And sometimes we don't know, and because there's limited data, and it's appropriate to say that the impact is unknown. So here are, on the left-hand side of the screen, I've included the required sections of labeling of the full prescribing information in physician labeling format, as well as the subsections in Section 12, the clinical pharmacology section of labeling. So the guidance creates a new dedicated subsection called immunogenicity, subsection 12.6. So here, this is really the home section for immunogenicity information. I think you could think of it analogously as the pediatric use subsection, subsection 8.4. Our understanding is that pediatricians go to subsection 8.4 for all pediatric use information, and that subsection may cross-reference other sections or subsections of labeling, but that's the home section. So it's similar to the pediatric use subsection 8.4. In the use in specific populations section, section 8, we created subsection 12.6, the immunogenicity subsection in the clinical pharmacology section of labeling, to be the home section for immunogenicity information. So healthcare practitioners can go to this subsection to find the relevant information. I should say that if there are some labeling that has already a subsection 12.6, and when folks update their labeling to be consistent with the guidance recommendations, that information would be moved to other subsections, potentially subsections 12.1 to 12.5, or maybe an additional subsection 12.7. I'll also talk during this presentation about other clinically oriented sections of labeling, sections 5, 6, and 14, and how and when immunogenicity information is incorporated in those sections. And to the earlier point, if there are clinically insignificant ADA, all the information, with rare exceptions, would be located only in the immunogenicity subsection, 12.6. So for the folks out there that are structured product labeling developers, we have created a new LOINC code. LOINC is also known as the Logical Observation Identifiers Names and Codes. So this is an SPL code that SPL developers use for structured reporting. So when immunogenicity information is incorporated into 12.6, when the SPL file is developed, they can include this code. So in the future, you could use labeling databases such as FDA Label or perhaps DailyMed to find immunogenicity information of labeling across many labeling. So the guidance talks about several principles about incorporating immunogenicity information in labeling, when to and how to do this, and there are three basic principles. One is to determine the adequacy of the methodology of the anti-drug antibody detection, whether data is sufficient to draw clinical conclusions, and finally, the determination of whether ADA may or may not have had clinical significant effects. So the focus of the presentation is the immunogenicity subsection. When the guidance is finalized and labeling is, when all labeling is updated to be consistent with the guidance, that's labeling therapeutic proteins and also small molecules that have immunogenicity subsection, immunogenicity assessments. Essentially, all such products will have an immunogenicity subsection 12.6. So this is a situation I'm describing on this slide and the subsequent slide when the methodology is inadequate. So if the methodology for the submitted immunogenicity evaluation is inadequate, which precludes the assessment of the set of ADA, the guidance recommends including this statement or a similar statement in the immunogenicity subsection to let folks know that there's insufficient information to characterize the ADA response. Now, most of the guidance, and in the majority of the overwhelming majority of situations, will be this scenario when the methodology is adequate. And I'll discuss in the subsequent slides with relevant examples about how to incorporate information in this subsection when the methodology is adequate. So I broke it down into several different elements. There are up to 4 different elements to include in the immunogenicity subsection when the methodology is adequate. The first thing to do, we recommend, is to include the immunogenicity clarification statement, and we recommend that the statement proceed come before the immunogenicity incidence. It's interesting in the article that I referred to earlier where we assessed 71 products that were approved, novel products approved in CDER, about 35% of the time, the immunogenicity statement did not proceed the data. But for a consistent approach across all labeling that have immunogenicity assessments, we would like this statement before, and it basically lets the healthcare practitioner know. I think it's a little bit about what Daphne was talking about, that there are different factors. There's drug factors, there's differences in dosage regimens, patient factors, concomitant medications that you really can't compare. It's really not meaningful to compare ADA data in one labeling compared to labeling across another biological product. Even the comparison is likely not to be meaningful if you're comparing biological products in the same class. So that is just basically a statement that proceeds the data to inform healthcare practitioners. Next, number 2, the recommendations are to include ADA data. Now, we recommend including ADA data whether the findings are positive, meaning that there are clinically significant ADA, whether the findings are negative. And then we also recommend including ADA. We recommend defining the duration of exposure. Usually it's the time period where you have sampling of ADA and during exposure to the drug. If you have data on NAB, neutralizing antibodies, the guidance also recommends doing that. So that's number 2. Number 3, if we have data, we're recommending providing data on ADA effects on pharmacokinetics or pharmacodynamics. As one of the commenters mentioned, it's most likely going to be ADA effects on PK. If so, we would include that under a specific heading title called Anti-Drug Antibody Effects on Pharmacokinetics. Previously, this information would go into the pharmacokinetics subsection, subsection 12.3. Now we're consolidating all the immunogenicity information in section 12. So if you have immunogenicity information, it should go into 12.6, not 12.3, and not 12.2. And finally, number 4, if you have PK or PD effects on safety or effectiveness, you should have a brief statement here about that, but the main concepts about adverse reactions, clinically significant adverse reactions, or subgroup analyses based upon immunogenicity with respect to effectiveness goes into the other clinically oriented sections of labeling. So I'm going through several different examples to illustrate some of these concepts. So example number 1 is a situation where the ADAs are clinically significant, meaning that there's an effect of ADA on safety and or effectiveness. And again, all these situations are when the methodology is adequate. So number 1, again, the 4 elements. Think about the 4 elements. Include the immunogenicity clarification statement to help healthcare practitioners interpret the data below. You can't make, it's not meaningful to make comparisons across labeling. Number 2, include the data. As mentioned earlier, we would like to have the treatment duration that this was assessed, and we're assessing immunogenicity in the drug-treated patients. Number 3, include ADA effects on pharmacokinetics under the specific heading, as you see there. For the purposes of this presentation, I'm highlighting in red, it's also potentially pink, some potentially concerning findings about immunogenicity. Yellow will be information that is unknown, and green are reassuring data that there less of a concern about immunogenicity impact. So this is pink, red, and what you're seeing here is that there's lower exposure in drug-treated patients who develop ADA compared to drug-treated patients who did not develop ADA. And then finally, in number 4, we're including a statement about how that affected effectiveness and or safety. And here is a brief statement, but again, the details of this will be in other clinically oriented sections of labeling. So that's example 1. Example number 2, this is also clinically significant ADA. And this is number 1, again, include your immunogenicity clarification statement. Number 2, include your data, again, over a defined period of time. Here is 26% of drug-treated patients develop ADA. Number 3, we include a brief clinical, the effects of ADA on safety. Notice here there's a split. There's a different effect of ADA on safety compared to ADA effect on effectiveness. So again, pink, red, there's potential concern for the purposes of this presentation that there was an associated ADA and a higher incidence of hypersensitivity, adverse reactions. However, with respect to effectiveness, that has not been fully characterized. So think, as we know, we think sometimes efficacy separately than effectiveness differently than separately from safety, and you could do that when you're developing this incorporating immunogenicity information in labeling. Now I'm going to turn my everyone's attention to a slightly different scenario. These are it's insufficient data to determine the clinical effects of ADA. And this is a situation when ADA percentage was low, and we don't know if ADA is clinically significant. Again, number 1, include the immunogenicity clarification statement. Number 2, include the data. And in this scenario, only 1% of drug-treated patients developed immunogenicity. Notice in the prior example, I think we had 26% of patients developed ADA. However, in that scenario, it was not clinically significant. That was the determination. So there was discordance between the percentage. You can't just look at the percentage of incidence. It's really how ADA affects safety or effectiveness. It's really the impact. And then later it's the management, if applicable. So again, going back to this example, only 1% of drug-treated patients develop ADA. And here we're having our effect statement, and we're saying because the low occurrence of ADA, the effects of ADA on pharmacokinetics, pharmacodynamics, safety, and our effectiveness is unknown. And again, the yellow is an unknown statement for the purposes of the presentation. Let's go to example number 4, still sticking with the category of insufficient data to determine the clinical effects of ADA. This is a slightly different situation where we have made a determination that ADA affects pharmacokinetics. However, it's unknown if the PK effects are clinically significant. Again, number 1, include your immunogenicity clarification statement. Number 2, include your data. And here, 5% of drug-treated patients developed ADA. Number 3, include your ADA effects on PK. And here we see a 20% lower exposure in patients who developed ADA compared to those that did not. However, we don't have sufficient data to determine whether the ADA-associated PK effects reduces effectiveness. And so you're telling, not just providing the ADA effects on PK, but we're trying to provide a healthcare practitioner some more useful information. And sometimes we don't know. And in our view, that's better to say that than to be silent. Okay, now we're switching scenarios. This is a situation when we have sufficient data to determine that the ADA that we observed are not clinically significant, meaning that there's no clinically significant effect, particularly on safety and or effectiveness. So number one, include your immunogenicity clarification statement. Number two, include your data. Here, 26% of drug-treated patients developed ADA. And number three, and I think this was the scenario I was talking about before, I was referring to before, I think I said it was the previous slide, but it was the subsequent slide. But anyway, even though 26% of drug-treated patients developed ADA, there was no identified clinically significant effect of ADA on PK, PD, safety, and or effectiveness over that treatment period. So again, that impact statement is important to tell the healthcare practitioner. The last example in subsection 12.6 is also based upon a clinically significant ADA. This is a situation when we have determined that ADA affects PK, but the changes in PK are not clinically significant. Again, number one, include your immunogenicity clarification statement. Number two, include your data. This is 5% of drug-treated patients developed ADA. Number three, here we have ADA effects on PK. And here, 10% of drug-treated patients who had ADA formation during the treatment period developed, well, I should say, sorry, that for the patients, drug-treated patients that had ADA had a 10% lower exposure compared to the drug-treated patients that did not develop ADA formation. So there was a lower exposure, 10% lower exposure in the patients who developed ADA during the treatment period. However, the determination is that these PK changes, 10% lower, was not clinically significant, was not identified to be clinically significant. Okay, moving on to a different section, section 6, the adverse reaction section. So, although, as you saw before in the review of 71 labeling novel drugs approved in CDER that had immunogenicity assessments, 98% of those labeling included immunogenicity information and labeling. If there are no clinically significant effects of ADA on adverse reactions, and in almost all cases, if the clinical effects of ADA on adverse reactions are unknown, there's going to be nothing in section 6 about immunogenicity. All that information will be moved to subsection 12.6. There is an exception for what we're calling narrow therapeutic index drugs, but in general, section 6 will not include information on immunogenicity. Now, this is a situation when we have ADA affects safety, ADA affects adverse reactions, and typically, as you know, this is likely going to be due to effects on hypersensitivity reactions and or injection site reactions. That's typically what you're likely going to find. And if that's the case, we recommend including the information under a specific heading, immunogenicity, anti-drug antibody-associated adverse reactions. And here you're showing in red, pink, that there was a higher incidence of hypersensitivity reactions in drug-treated patients with antibodies compared to those that did not have antibody formation during the specific treatment period. 9% versus 2%, and one of the patients who developed anaphylaxis had anti-drug antibodies. So as you can see here, the concentration is really about adverse reactions. It's not, you don't see immunogenicity incidence. The incidence is in subsection 12.6. As you can see, the cross-reference here to 12.6. If there are clinically significant adverse reactions related to ADA, that may be included in section 5, and here you see a cross-reference to section 5. This is the uncommon situation. If you have clinical effects on ADA on adverse reactions are unknown, and the situation is where minimal concentration changes for the drug may lead to serious toxicities, the recommendation and the guidance is to include this statement under the heading immunogenicity, unknown clinical effects of anti-drug antibodies. There are insufficient data to evaluate the effect of ADA antibodies on adverse reactions. Okay, switching now to the clinical studies section of labeling, section 14. So as I stated earlier, typically you're not going to have immunogenicity information in labeling. These are the scenarios where there are no clinically significant effects of ADA on effectiveness. You've done a subgroup analysis by ADA formation, and you didn't see any differences in effectiveness. You would not include anything in section 14. Similarly, if the effects of ADA on effectiveness are known, for example, if there's a low incidence of ADA, you're not going to include anything in section 14, and there is that exception of narrow therapeutic index drugs that we'll talk about later. So if you do have a situation when there are clinically significant changes in effectiveness associated with ADA formation, you would include that subgroup analysis in section 14. So let's go through an example. This is a psoriasis, a fictitious psoriasis example. What I'm presenting here is typically what you would see in a section 14, right? You have a description of a slight description of the study. You have a description of the primary efficacy endpoint. In this situation, it's the PASI 75. It's a reduction in the PASI score of at least 75% over a defined period of time. And in here, the results of the study, 89 versus 10% of drug-treated and control-treated patients achieved a PASI 75, which looks wonderful for the product. It was a success. Looks like there was a big differential with respect to efficacy in the drug compared to the control, 89% versus 10%. And notice here, there's no mention of immunogenicity. So this is presenting the overall results. This is the typical standard approach as per our clinical study section labeling of guidance. Now here, the second, later down in the clinical study section of labeling, one would include a subgroup analysis of effectiveness based upon ADA response. So here you're looking at, if you look at drug-treated patients who develop ADA during that defined treatment period, 50% achieved a PASI 75, that's the primary efficacy endpoint, compared to 90% of drug-treated patients that did not develop ADA. So here you could see a drop-off of effectiveness, still much better than control that we saw before, but lower than compared to those that did not develop ADA. Here, we do not report the ADA incidence. You notice the cross-reference to subsection 12.6. And there could be a cross-reference to also the warnings and precautions section if there are clinically significant information about immunogenicity in that section. So here is the uncommon situation when you have a product for which minimal concentration changes may lead to the loss of effectiveness. The recommendation is to include this statement or a similar statement after the description of the results of the studies. You would also include a cross-reference to the immunogenicity subsection of labeling that provides more information about the data. The last section of labeling for discussion is the warnings and precautions section. As you know, the warnings and precautions section must contain clinically significant adverse reactions or risks. And here, If we do have clinically significant adverse reactions or risks from ADA, this information would be included in the in the warnings and precautions section of labeling. This is a fictitious example. What you see here is what you would see with the typical warnings and precautions section of labeling. Here, these are severe hypersensitivity reactions, including anaphylaxis. There's no mention of immunogenicity. It's just talking about hypersensitivity reactions. Then you would include information with respect to the relationship of clinically significant adverse reactions or risks with respect to immunogenicity. In this situation, there was one patient who developed anaphylaxis who also had ADA formation. And we're cross-referencing to other sections of labeling for more information. And finally, as recommended in the warnings and precautions section of labeling guidance, we recommend an action if known, and if someone develops severe hypersensitivity reaction, the recommendation is to discontinue the drug. And finally, although it's uncommon, we may include information about lack of efficacy in the warnings and precautions section of labeling. So we can include information about clinically significant changes in effectiveness associated with ADA in the warnings and precautions section. So for the final concept in this presentation is updating immunogenicity information in labeling. So if we have new immunogenicity information that could affect prescribing decisions or clinical management, we recommend applicants submit a supplement with proposed updating labeling to include that information. So for example, if post-marketing there is an occurrence of, let's say, injection site reactions, and those reactions are associated with ADA, in a post-marketing study, and there's a differential incidence potentially in those that had ADA formation compared to those did not, we may want to include that that injection site reaction information in the adverse reaction section, and we would recommend the applicant submit a supplement to update that data in the labeling. Now, the recommendations and the guidance, when the guidance is finalized, we recommend that applicants propose labeling updates to be consistent with the format and organizational recommendations in the guidance. And for example, the next plan prior approval supplement. And we're doing this to try to have some consistency, as I noted before, in our assessment before over the 5-year period, we've had a somewhat inconsistent approach to incorporating immunogenicity information labeling, and that's not surprising when we don't provide formal recommendations. We have some inconsistency, but we are recommending that applicants, when the guidance is finalized, update their labeling to be consistent with the recommendations and the guidance in the next plan supplement. And certainly in the draft stage, applicants can voluntarily update their labeling to be consistent with the recommendations in the draft guidance. And we think that applicants are likely to do this given the concepts that we discussed in this presentation. So finally, as I stated before, this is a link to the docket. We welcome your comments. As I stated earlier, we received 2 thoughtful comments as of last night. We will look at your comments prior to finalization of the guidance. So now we're on to the challenge questions portion of the presentation. And first of all, I will read the question, and then everyone could read the answers and answer the questions. So which of the following statements is inconsistent with FDA's recommendations for the immunogenicity subsection 12.6? And I'll give folks time. Great. It seems like the majority of folks got the question correctly. So basically, we do want to include immunogenicity data and summarize the studies. We do want to, if known, identify the ADA effects on pharmacokinetics. We do want to provide the duration of exposure to the drug and the time period for sampling ADA. And if known, we want to provide neutralizing antibodies. As I stated earlier, we do recommend keep maintaining the subsection 12.6, even if there's insufficient data. We do think it's helpful to provide the information and that statement that there are insufficient data to make a clinical determination of the significance of ADA. Next question. Great, let me read the question. What anti-drug antibody information should appear in the adverse reaction section, section 6? And I should say these are all recommendations from the immunogenicity labeling guidance. Okay, great. I think we have a reasonable number of responses. The answer is actually, I think that's D. The answer is adverse reactions associated with ADA hypersensitivity reactions. So, with respect to ADA incidents, the guidance recommends only including ADA incidents in subsection 12.6 cuts. But so the adverse reaction section really concentrates on adverse reactions. We're looking at the adverse reactions in drug-treated patients who developed ADA formation compared to compared to the incidence of adverse reactions in the folks that did not have ADA formation. So it's really about adverse reactions. Remember, adverse reaction section is about adverse reactions, and the data, immunogenicity data, is in subsection 12.6. Next question, please. Okay, great. What immunogenicity information should appear in section 14, the clinical studies section? Okay, great. I think we have a sufficient response. The answer is include subgroup effectiveness analysis by ADA formation when ADA associated with clinically significant changes in effectiveness. So as stated earlier, ADA incidents will only be included in subsection 12.6. We don't want to routinely include subgroup efficacy analyses. We only want to include it when you see a potential concern that there is lower effectiveness in patients who developed ADA compared to patients that did not in the drug-treated group. But adverse reactions information, adverse reactions is safety information. Generally, safety information is section 6. Hypersensitivity reactions generally is in section 6. Okay, next question, please. What immunogenicity information should appear in the warnings and precautions section, section 5, if known? Okay, that's great. I think most folks got this correct. All of the above. All these items are actually in the warnings and precautions section of labeling regulations, as well as the warnings and precautions section of labeling guidance that was finalized in 2011. Next slide, please. Next question, actually. Oh, great. That's the end of our questions. Oh, no, the last question, sorry. Immunogenicity information in the labeling must, should, or may be updated when? And that's great. Almost everyone got that correctly. Great job. We recommend the immunogenicity information labeling. Well, it must be updated when it could affect prescribing decisions. When the guidance is finalized, the recommendation is to update the labeling to be consistent with the recommendations in the immunogenicity labeling guidance in the next supplement. That's great. And now we're
Thank
going
you
to
both.
turn our attention. Oh, hi, Ray. We're going to turn our attention to Ray, who's going to start with the question and answer section. Thank you, Ray.
Thank you both for the great presentations, and we're going to move into our Q&A session. And just remember, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Looks like we have some questions coming in right now. All right, while they come in, vetting those. And the first group of questions looks like it'll go to Dr. Gwen. And here is the first question for Dr. Gwen. Can you comment on the type of small molecule products where immunogenicity should be considered?
Thanks, Ray. So an example that was provided in the presentation was peptide products, which are polymers composed of 40 or fewer amino acids. These products can share similar characteristics with other protein products that are defined as biological products, and immunogenicity can be a concern for these types of products. In the published landscape analysis that Eric mentioned of FDA labeling for CDER approved products, the types of small molecules that were identified in that landscape analysis to have some immunogenicity information in labeling were peptide products, oligonucleotides, and siRNA products. Sponsors are encouraged to discuss this assessment of their product's immunogenicity risk and how it could inform the clinical immunogenicity assessment early in their development program. And there may be other novel types of small molecules that emerge that could have a possibility of immunogenicity risk. So bringing that to the agency early is important. Thanks.
Thank you for responding to that question. Looks like we have another question just came from Dr. Gwen, and here is the question. What are some of these strategies sponsors should consider when planning the assessment of immunogenicity during product development?
Yeah, so as I mentioned, FDA has several guidances that provide the agency's current thinking on immunogenicity assessment and assay development and validation. These are invaluable resources along with consulting the agency's relevant review division early in the development of the product because each of these do have a unique risk assessment. So this would just ensure that your study design, your sample collection, and your analysis will be robust enough to assess immunogenicity incidence and any potential impact on immunogenicity may have on PK or safety or efficacy. So these aren't a one-size-fits-all approach typically. for each product, there are unique considerations.
Thank you for responding to that question. Looks like we have one more question for Dr. Gwen, and here is the question. You mentioned that there may be a range of immunologically related adverse clinical events for different products and provided a couple of examples. Can you discuss any additional cases where a product formed ADAs but had no clinical effect?
Thanks for that question. So I'll go back to that published landscape on immunogenicity information and FDA labeling for CDER approved products. So the majority had immunogenicity information and incidence information in their labeling. And when we looked at that information, it described an immunogenicity impact. There was no observed immunogenicity impact in about a third of those products. A third of those products didn't provide any clinical impact information in their labeling, so there was just no information to assess. And then the remaining products had language that indicated that they either had an observed, potential, or unknown clinical impact on immunogenicity. Some of these immunologically related clinical adverse events can be really rare, such as the PRCA example that I gave. These rare events can be potentially serious and pose a great safety risk. So, and they may not necessarily be detected in the pre-approval clinical testing based on the size of the population that is exposed. May not be large enough to assess these rare events. So the agency does encourage sponsors to consult the agency to discuss their proposed plan for post-marketing safety monitoring, and then also updating their immunogenicity information and labeling post-approval, just to ensure that the drug is being used safe and effectively. Thank you.
Thank you for responding to that group of questions. We'll now move on to Dr. Brodsky. Looks like we had several questions coming for Dr. Brodsky, and here's the first question. When we move immunogenicity information from the adverse reaction section to the immunogenicity section, will any information remain in the adverse reaction section?
Ray, that's a good question. So in that scenario, one would move the, number one, the immunogenicity clarification statement, ADA incidence data, if you have NAB data, neutralizing antibody data, and as well as ADA effects on pharmacokinetics. That information would be moved from section 6, that sometimes that information appears, to subsection 12.6. In most cases, there'll be no information in the adverse reaction section. However, there will be several cases that certain ADA information would remain, specifically information where you had a higher incidence of ADA adverse reactions associated with ADA. That information would remain in the adverse reaction section, and one would cross-reference the adverse reaction section to the immunogenicity subsection, subsection 12.6 in the clinical pharmacology section.
Thank you for responding to that question, Dr. Brodsky. We have a few more questions for Dr. Brodsky, and here's the next question. If ADAs were not found to be clinically significant, can section 12.6 be omitted?
That's a good question. We have, although we have omitted several sections and subsections of the labeling, for example, we do typically remove the drug abuse and dependence section of labeling, section 9, if there is no information about abuse, addiction, misuse, tolerance, dependence that's relevant, for example, if it's a biological product. But in this case, our recommendations is to maintain subsection 12.6, primarily because we would include information for a healthcare practitioner that basically states that we have assessed immunogenicity and we have not found it to be, we have not identified ADA effects on safety or effectiveness, and we think that will be helpful to healthcare practitioners.
Thank you for responding to that question. We have one more question to Dr. Brodsky in this round. Here's the final question. In slide 44, you show an example where hypersensitivity reactions are described under a specific recommended immunogenicity heading in the adverse reaction section. However, in many cases, hypersensitivity reactions may not be associated with many observed ADAs. How would this be conveyed?
Hi, Ray. That's actually an excellent question. As the comment noted, frequently we do have hypersensitivity reactions and there's no association with ADA. One would develop the adverse reaction section. Normally, potentially those hypersensitivity reactions would be included in the common adverse reaction table, potentially would be listed low after the common adverse reaction table. So it really depends upon the data. Sometimes in the situation that you may have hypersensitivity reactions associated with ADA and some not associated with ADA. In that scenario, one could potentially have two different headings. You could potentially have a heading of hypersensitivity reactions and another heading that focuses on immunogenicity. As the guidance recommends, immunogenicity, anti-drug antibody-associated adverse reactions. It really depends upon the data. It's a really good question. Thank you.
Thank you for responding to that question. We have some questions that just came in for Dr. Gwynn, and here is the first question. Can you provide an example of when immunogenicity has been extrapolated?
So I'll start by saying that immunogenicity can be unique for each product, and each risk assessment considers those patient and product factors. There are certain product classes or product modifications that may have shown immunogenicity impact on safety or efficacy, and this may be labeled to highlight that potential risk for a particular product class or product modification.
Thank you for responding to that question. We'll go on and have another question for Dr. Gwynn. We've got a couple more. Here's the next question. How does the FDA define therapeutic proteins?
So FDA's definition of biological product, they recently changed this definition, the statutory term protein, and under this final rule, the term protein means an alpha amino acid polymer with a specific defined structure that's greater than 40 amino acids in size. CDER-regulated biological products can include monoclonal antibodies, proteins that are intended for therapeutic use, such as cytokines, enzymes, and other novel protein products.
Thank you for responding to that question. We have one more question for Dr. Gwynn, and here's the question. In footnote 23, it states that the immunogenicity studies may not be required for some biological products. Can you give some examples of what types of biological products may not require such studies?
Yeah, so the sponsor should always consult the appropriate review division to determine their strategy for assessing immunogenicity. There may be cases for certain ophthalmological products, as the eye is an immunologically privileged site, that may not need an ADA assessment. But the final determination would be based on that overall immunogenicity risk assessment, considering those patient and product factors for the individual product, specifically, looking at what the indication is and how the product will be used. So thank you.
Thank you for responding to that group of questions. We're going to move now back to Dr. Brodsky. We have some more questions that came in for Dr. Brodsky, and here's the next question. Does the development of the immunogenicity section follow the same format recommendations for vaccines?
Hi, Ray. That's a good question. So the guidance, for vaccines, an immunological response is really part of the purpose of the vaccine. So it's to induce a specific immune response to prevent a disease. So that's a positive thing or desirable immunogenicity. And the guidance really, the focus of the guidance is undesirable immunogenicity, as Dr. Gwynn mentioned earlier, so the guidance does not apply to vaccines.
Thank you for responding to that question. We have a couple more questions for Dr. Brodsky. And here's the next question. For newer generations of biologics within a class, if all the previous older products in the class have immunogenicity data summarized in the adverse reaction section, should the new product also do the same for consistency? If not, there will be an inconsistent format organization across products of a class.
Hi, Ray. That's a good question. So we recommend when the guidance is final to update one's labeling, biologics, for example, labeling to be consistent with the recommendations of the guidance, and eventually, so all biologics will have a subsection 12.6. So again, this is the labeling is a communication tool for healthcare practitioners, and it is desirable to have a consistent location, a home subsection, where people can find this in a consistent location in the prescribing information.
Thanks for responding to that question. We do have another question for Dr. Brodsky. What happens to the labeling if a biological product is initially marketed with an inadequate immunogenicity evaluation and subsequently additional data is submitted to demonstrate the adequacy of the methodology?
Good question, Ray. So frequently assays are developed over time, sometimes after approval of the product. So if the methodology is determined to be inadequate at the time of approval, and in the post-marketing space, it's developed further and it's determined to be adequate, that statement that we discussed earlier would, in subsection 12.6 would be removed, and one would populate the immunogenicity subsection appropriately. One would include the immunogenicity clarification statement, the ADA data, and then there would be an assessment of ADA effects on PK, PD, safety, and or effectiveness. And if appropriate, information about ADA effects on safety would be included in section 6 and or 5, and ADA effects on effectiveness, although rather uncommon, would be in section 14. Thank you.
Thank you for responding to that question. A couple more questions just came in for Dr. Brodsky, and here's the next question. What are your recommendations on how to include immunogenicity information in biosimilar labeling?
Ray, that's a good question. As you know, we have over 30 biosimilar and biosimilar interchangeable products approved in the United States. We're very happy about that. Provides patients, healthcare practitioners, additional options when appropriate. The recommendations, we have a 2018 biosimilar labeling guidance that describes how to incorporate information in biosimilar labeling, and that essentially states that the biosimilar labeling essentially mirrors the reference product labeling, except for nomenclature differences, except for if the biosimilar is approved for less than all the conditions of use of the reference product. So when you're developing a biosimilar labeling or biosimilar interchangeable labeling, one would mirror the data that's included in the reference product labeling. So the data would be from the reference product. With respect to the organizational changes, moving information from section 6 to subsection 12.6, it would only follow after the reference product labeling. So for example, if the reference product labeling has not had a chance to update its labeling to be consistent with the recommendations in the immunogenicity guidance, so that the data is in section 6, the biosimilars data would still, the data, immunogenicity data would still be in section 6. However, when the reference product updates its labeling to be consistent with the immunogenicity labeling guidance, the biosimilar subsequently would submit a supplement and follow the reference product and move its information to be consistent with the reference product labeling.
Looks like we have just a few more minutes. We're going to try to squeeze in one more question with Dr. Brodsky, and here is the final question. For products with an ADA clinical impact on efficacy and safety, the impact on efficacy is described in Section 14, and the impact on safety is described in Section 6. What information should be included in Section 12.6?
Ray, that's a good question. When we develop labeling, we go back to the principles of each of the sections of labeling. And the principles of Section 6 is really adverse reactions. The principles of Section 14 is effectiveness. And subsection 12.6 concentrates on immunogenicity data. There will be, so if you're having your subgroup safety analysis by ADA formation, that should be in Section 6. If you're having a subgroup effectiveness analysis by ADA formation, that should be in Section 14. Again, this is the scenario when there's a clinically significant impact on effectiveness and or safety. When there's not, one would not include information in Sections 6 generally or 14. Subsection 12.6 really concentrates on the data as well as ADA effects on PK and or PD if we have that. There could also be some information, a summary statement about PK effects on effectiveness and or safety, and there's a cross-reference for the clinically oriented sections of the labeling. But you go back to the fundamentals of the principles of those sections of labeling.
Well, that's just about all the time we have for questions. We can give an opportunity for Dr. Brodsky and Dr. Gwynn if they would like to say any closing comments before we finish with our housekeeping information.
Thanks, Ray. I really appreciate everyone's participation in this webinar. We received your questions. Not only will review the questions that are submitted to the docket, and please submit questions there, but we will also review all your questions. I'm sorry we didn't get a chance to answer all of your questions, but we will consider those questions before we finalize the guidance. And just some take-home points here. As you know, we created this dedicated news subsection 12.6. This is the home subsection for immunogenicity information labeling. And the purpose of the guidance, the main purpose here, is to distinguish between the products that have clinically significant immunogenicity, for example, ADA effects on safety, and that would be in the adverse reaction section of labeling, from those that don't. And those that don't, the immunogenicity information will be concentrated in the dedicated subsection. Thank you, and we really appreciate your participation, your questions, and as well as your comments to the docket.
Well, we certainly want to thank our speakers with a huge thanks, and for their very informative and timely talk, and also for responding to numerous questions that came in. We do have a few closing reminders with our attendees. If you'd like a certificate of attendance that may be used in support of RAPS, SOCRA, SQA, and ACRP credits, please email your request to cdersbia@fda.hhs.gov. Certificates of attendance will be emailed until the April 19th deadline. We apologize in advance, but we're unable to make exceptions to that April 19th deadline. So you'll be able to request your certificate of attendance for 2 weeks until April 19th. Additional tutorials are available through fda.gov/CEDARLearn. And to summarize, for our physicians, pharmacists, and nurses claiming the 1.5 continuing education credits, you have 14 days from the last day of the activity to log in, complete the required CE evaluations, and attest to your attendance to claim credit. So be sure to complete the CE evaluations on or before April the 19th. SBIA will issue the CE claim code to all attendees later on today. And be sure to complete once again, April the 19th, the deadline for those CE session evaluations. On behalf of CEDAR Small Business and Industry Assistance, we hope you enjoyed today's presentation and found it helpful. We look forward to your presence at future webinars, which will be advertised on our website at fda.gov/CEDARSBIA. Thank you.